Up next, we have Rachel Washburn from the Kentucky Geological Survey. Her research will take us to deep underground, where she's exploring Kentucky caves, exploring Kentucky cave for the search of new medicines. So please welcome Washburn.
All right, thank you. Can you hear me? Is the mic working? Okay, good. All right, so I'm Rachel Washburn, and like you said, I'm going to talk to you about why we're screaming into the void at the Kentucky Geological Survey. So we're doing it to do bioprospecting, and I feel like bioprospecting is just a really fancy word for saying we're looking in biology to try to find stuff that we want to use, particularly things for medicine or for different industrial advancements. My work specifically focuses on microorganisms. So instead of looking at plants, I'm looking at the little bugs that are existing within these caves. And I'm looking to see if they're making any kind of novel metabolite, which is just a chemical that we can use and turn that into something like anti-inflammatories, anti-cancer therapies, and antibiotics. Basically, we're just molecular treasure hunting in extreme places. So why does bioprospecting matter? We are in the middle of an antibiotic-resistant crisis where we're honestly running out of a lot of effective antibiotics. So there's a great need for us to come up with more ways to treat against these infections, especially when we're coming up with things like the antibiotic-resistant UTIs, antibiotic-resistant plague that's coming up. So there's a need there. There's always a need for new cancer therapies, and some of the extremophiles that we see, these bacteria and microbes living in these extreme environments, might make compounds that we can utilize to treat different cancers. Bioremediation, there have been microbes that have been identified that can help clean up pollution, and rare earth elements, which are just those elements that we use in things like lithium batteries to keep our electronics working. Microbes have been known to make chelating agents, which are just enzymes that can pull metals out of the environment, so we can use it there too. Lots of uses for bioprospecting. So the knowledge gap that we're facing right now is we don't really understand how geology and geologic conditions are shaping these microbial responses in places like caves. And the very things that the microbes are making, the metabolites, the enzymes, the bioactive compounds, maybe those geological processes are key in microbes producing that. So we're hypothesizing that in places like Kentucky caves, that we're going to see geologic processes are affecting microbes to make these compounds. All right, so the way that we do this is we have to go underground. We have to go into the Kentucky underworld, and specifically, Kentucky is well known for caves, right? Especially Mammoth Cave, and that's the exact area that we're going. So within the Mammoth Cave region, we're sampling 10 different caves. And one thing that we're doing that's kind of interesting is we're not just sampling like national park caves, we're looking at 3 different cave types. We're looking at protected caves, so caves where people coming in, they're really monitored, they're looking at who's coming in, making sure nobody's touched anything, and trying to keep the cave more wild. We're looking at scientific research caves, where the only people that can come in are scientists doing research, but we're also looking at show caves. So these are the caves that are privately owned, they're not managed by any park service, and honestly, tourists come in and they're touching everything. People have basically become part of these cave ecosystems. So we're sampling in these 3 different areas across 10 different caves to see what we find. And when we go into caves, the first thing we do is field research. So I go in, we make sure we have all our permitting, and permitting is the part that literally takes the longest. And then once we go in, we collect things like sediment, we take swabs and we swab the cave walls, and sometimes they let us swab the stalactites, which is pretty cool. We get water samples from cave pools, but also we'll stand there under a dripping stalactite and wait till we get water. We're trying to collect both types to see what we're finding. We get rock samples, geology, rocks. We make sure to collect those too. And we get atmospheric data. What's the light looking like, the temperature, the pressure. We collect all these under the most sterile technique we can do in an environment like this. So we glove up, we use ethanol and bleach, but we keep it restricted to an area so we're not affecting the cave ecosystem. And we put everything separately. They're all in their own separate little sample collection tubes. We immediately stick them on dry ice, and once we get back to the lab, they go into our freezer. So we're trying to keep these microbes and metabolites viable for as long as we can. In the lab, we do 3 different types of experiments: molecular biology, geochemistry, and then we look at all of our environmental metadata. So for molecular biology, what we're looking at in our samples is what microbes are there. We can also look at gene expression, what genes are being expressed and what are they doing. We look at the different cell structures, which can make targets for new antibiotic therapy. And a big one is we're looking at metabolites, which is, again, that chemical arsenal these microbes are using that we could potentially take and use to make things like new antibiotics. Geochemistry-wise, we run our samples to figure out the basic chemistry. What are the elements within our samples? What are the minerals that these samples are made up of? And we do trace chemistry. So we're looking for things like rare earth elements, like heavy metals. We're looking for things like nitrates. What is in this environment that the microbes are living in? In our environmental metadata, again, we have temperature. We look at light, we measure water, and we also measure things like methane and carbon dioxide. So what I do is very interdisciplinary. I'm at the intersection of biomedical sciences and geological sciences. So we do a lot of interdisciplinary integration. Our microbial diversity, this is like guests attending our party. So we're looking at who's attending the party and how many people are showing up, and maybe how this party is different from a party further down in the cave. Geologically, this is like our venue. So what's on the stage? What's at the event? What are the party supplies that are available? And then we look at biochemical processes. These are the activities. What did everybody bring to the party? What are they doing? What's happening at this party? So we look at all these separately, and then we look at how they integrate together. And this is our integrated systems analysis. This is our full event. So we're looking at what is actually happening at this party, how's it playing out, and how are the venue, the guests, and the activities all feeding into what we're seeing. And how can we take that and have better parties in the future? So far, our preliminary results are, first of all, cave access acquired. We have gotten access to 7 out of our 10 caves so far. That has been a full process. But we got access, and we've been going into these caves. We've extracted our microbial DNA. We're sequencing it right now to see what do the guests at our party look like. We're extracting metabolites, and we're running chemical and molecular networking to figure out what that chemical arsenal looks like. And our geological data, we're collecting that too, looking at what minerals are actually in this environment and how that compares to the different cave sampling sites. All this is helping us build these integrated cave profiles so we can put together that microbial, biochemical, and geological profiles to see what's going on in this big, messy environment in the underworld of Kentucky. We're also doing geospatial integration. So we are literally mapping microbes across the state of Kentucky. So all our cave data is being added to this map that we have. That's giving us this microbiome of Kentucky, and it's helping to reveal potential bioprospecting patterns. Can we identify hotspots that would be ideal for us to know where to go hunting for these molecular treasures? And this is also something to give us like a guided map. Instead of just shooting blind, we might be able to target what we're looking for and be more effective in our studies. This is also an open access platform because we collaborate with a lot of people. We're very interdisciplinary, and we want to encourage people to use these products so we can work together and develop these drugs. All right, so overall, my work, we do a very integrated approach: geology, biomedicine, biology. And we're looking at bioprospecting Kentucky caves specifically, which could be the new future of medicine to give us things like these potential breakthroughs: new antibiotics, anti-inflammatories, anti-cancer drugs. And we're putting Kentucky at the forefront of geospatial-based microbial discovery. So being at the Kentucky Geological Survey, we're in a unique point where we can actually survey a lot of the state. We don't have to worry so much about picking one specific site, and we can put ourselves ahead there and see more what the big picture is and see how we can apply that big picture to other locations and other studies. All right, thank you for listening.